Dihybrid cross. So this time we're going to be looking at two characteristics when we cross um, peas or whatever we're crossing. So this is an experiment that tracks the inheritance of two traits. So let's go over that as an example. So we might be looking at eye color and, I don't know, height, pretending that height is a one gene thing, or pretending that eye color is a one gene thing. I don't know. So anyway, your eye color, you might have big B for brown and little b for blue, and height, you might have big T for tall and little t for short, and that one might be, that person might marry someone with blue eyes who is short. And so what's going to happen in this mating? What kind of babies will you have? So you're tracking two things, both eye color and height in both of the organisms. And of course, these aren't really good examples because eye color is controlled by at least two genes and height is controlled probably by a whole bunch of genes. But anyway, each pair of alleles segregates independently during gamete formation. Now that's what Mendel said, and it was certainly true for all of the traits in peas that he looked at, but it's not necessarily true of all of your traits. It's certainly true of any traits on different chromosomes, or if they're far apart enough on a chromosome, then it's true for them. So we're going to just assume this is true for now, and we'll talk about when it's not later. So here's a dihybrid cross. Let's look at seed shape and seed color. So this is something that Mendel would have done. So the alleles are big R for round and little r for wrinkled, big Y for yellow and little y for green. And so here's your cross. You're doing heterozygous round and yellow and heterozygous round and yellow. And so the first thing that you have to do is figure out what gametes each one can make. So this one can make, it's sort of like you do FOIL in math first. It can make big R, big Y, outer, big R, little y, inner, little r, big Y, first, outer, inner, last. So little r, little y. So that's the FOIL method that you learned in math probably. And it really is the same thing. So these are all the all the gametes that this one could make. So maybe these are all the pollen. So notice that for each pollen, there's just one allele for each trait, because remember you separated out the two alleles when you did meiosis. And so same thing for the eggs, big R, big Y for one of them, big R, little y for another, little r, big Y, and little r, little y. So you got to make sure, here's a common mistake. Kids will write a gamete like this, and they'll write another gamete like this. And so you've got to tell me what's wrong with that. So the thing that's wrong with that is this organism is going to have two alleles for the shape, round or wrinkled, but no alleles for color, so it won't know what color to be. This one will have two alleles for color, which is too many, actually, and it'll have no alleles for the shape. So you want to be careful with that. So these are all the possible gamete combinations, and that's really I think, the hardest part of the whole thing. And that's what you'd have to do for an MCAS problem. Usually that's as far as you have to go with a dihybrid cross for them. For me, I'm going to make you do a little bit more. So you get your Punnett square, and this time it's a huge one because you have to write all of the pollen here and all of the eggs along here, or maybe the other way. I think I did blue for the pollen. So each gamete has one allele for each trait. So for the seed shape, there's one allele, and for the seed color, there's one allele. Same thing with this pollen. For the seed shape, there's one allele, and for the seed color, there's one allele. Remember that all the gametes from the male go on one side. I don't really care which side. And all the alleles for the female go on the other side. And again, as long as it's a different side from that one, it doesn't really matter. You want to make sure you're not putting a female allele here and then here, and you'll get it all messed up if you do that. So from here, you're just filling it in. This one's going to have two big R's, one from here and one from here, and two big Y's here and here. So let's fill that in. So yikes, so there's a lot to look at here. So the way you analyze this, I think you're probably okay with drawing that big R, big R, big Y, little y. So you're thinking of each of these pollens as fertilizing potentially each of these eggs. So these are the possibilities that you can get in the offspring. So this one is round and yellow. Round and yellow, even though it's a heterozygous one, this is the dominant one. Round and yellow here, don't worry about the little r because it's got a big R. Round and yellow, round and yellow, not, that one's not because it's two little y's. round and yellow, round and yellow, round and yellow. That one's not, that one's not, that one's not, that one's not. That one, oh, it's not. That one is. So to be round and yellow, that's 1, 2, 3, 4, 5, 6, 7, 8, 9. So 9 of them, or roughly 9 out of all of these, would be statistically round and yellow. How about round, and I think little y was green. Hopefully I have this right. So big big R and little y. So that's this one and this one, and that's it. So there's only 2 of those. And how about if they're wrinkled? Let's look for the wrinkled yellow ones. So that would be little r's with a big Y. So that's this one, and this one, and this one. 3. Oops, look, I missed one of the round green ones right here. Oh my gosh. I knew there should be another one. I just couldn't see it. Yay. Okay, so the last one, let's see, little r would be wrinkled, and little y would be yellow. And so, whoops, sorry, little y is green. And so for the little y, that would be just one of those. So you get this 9 to 3 to 3 to 1 ratio. So you see that all the time, 9 to 3 to 3 to 1 ratio. You get that when you have heterozygous organisms for 2 traits mating. So 9 to 3 to 3 to 1, that's the phenotypic ratio, because we're counting this really is the same thing as that, even though it's not. It's a genotypically different, but their phenotypes are both going to be round and yellow. And here's what they would really look like. Here's a round yellow one, and here's a wrinkled yellow one, and here's a round green one, and here's a wrinkled green one. So physically or phenotypically, that's how they look. So a test cross, remember with a test cross, you're mating an organism of unknown genotype with a homozygous recessive one. So unknown genotype and homozygous recessive. So the unknown genotype, that means you have a dominant trait and you don't know if it's homozygous or heterozygous. So in this case, you'd be doing it as a dihybrid cross. So here's an example. You have a, well, let's look at what these traits are. So little b would be blue eyes. So this one is little b, little b. So this organism is mating with this organism and they both have blue eyes. This one has a big C for curly hair. And this one has little c for straight hair. So this isn't necessarily the way it is in humans, but let's just assume that it is for a little bit. If you have a curly-haired person, you're not sure whether it's big C, or big C, little c. And so the possible gametes for this one are little b, big C, or maybe little b, little c. They might be all this way, or if this is big C, or that might be a little c. The only possible gamete for this one is little b, little c. So this is the possibility here, and this one we don't really know, right? So the possible results, if it's a big C, then that means all the organisms are blue-eyed with curly hair. Or if the organism has a little c, that means the chances are that half the kids would have blue eyes and curly hair, and the other half would have blue eyes and straight hair. So here's a summary of all of Mendel's laws. And the reason that we did dihybrid crosses is so that you can see an example of independent assortment. So dominants would be big T, and little t. If you have the offspring, they're all big T, and they're all tall. So it's showing that this is dominant over that. Segregation means that these things separate. So even though, so you've got a big T, and a big T, that means when you make gametes, one of them's big T and one of them's little t. Big T and little t. Okay, so when you do this, you get some offspring that are little t. And if things didn't segregate, then you wouldn't be doing that. You'd have some kind of blending or something. And what Mendel found is that no, these recessive traits, even though you don't see them, are still there. So that's what this means. And then the last one, independent assortment, that means when you have two different traits, like shape and color, these things assort independently. So you get that 9 to 3 to 3 to 1 ratio at the end. So the big R can go with the big G, or the big R can go with the little g. And the little r can go with the big G, or the little r can go with the little g. And so that was different. That was something that nobody had known before. And it's actually not true if you look at a chromosome and you have traits that are very, very close to each other, but it's true for all the other traits. Any trait that's on a different chromosome or far enough apart, it would be true for. Okay, Anna, can you say goodbye?
Goodbye.